Welcome to this. Very warm welcome to this Pulses Challenge DDD seminar. I'm Nathan from DDD at Scilab Lab. I'm very happy to invite Dr. Muthukumaran from Yale University. He's currently an associate research scientist in the Department of Chemistry at Yale University, and he is working together with Professor Craig Crew and Professor David Spiegel. With a strong background in chemical biology, he earned his PhD from Max Planck Institute from Germany. And he has very strong background in chemical biology, and now he works mainly on developing novel modalities for targeted protein degradations and also therapeutic discovery. And today's seminar title is From Serendipity to Design Principles, Molecular Glues as a Prelude to RIPTAC. So Dr. Muthu, thank you for joining us today. And so now I'll hand over to you, Muthu.
So I stop sharing now, and I will allow you to share, Muthu.
I hope you can hear me. Can you hear me? Yeah, perfect. Yeah, so thank you for the opportunity. My name is Muthu. I am a scientist at Yale University. I'm going to talk about molecular glues and RIPTACs today. I want to start with the disclaimer. I'm not affiliated to ALDA, which is the company founded RIPTACs, and whatever explanation and opinions I provide are all my own. So I'm going to touch upon three topics. One is the photo-caged control of protein interactions. That's my short gist of my PhD chapter, how it is relevant to RIPTACs. I want to touch base on that. I will talk about the evolution of molecular glues, and finally I'll touch upon the RIPTACs and the clinical data what we have from the RIPTAC story. So to begin with, that's my boss, Professor Leif Demelt. Leif was PI at Max Planck, and I was a PhD student. This is part of my PhD work. So what we did here is, I want to use a pointer. So this is a cell, and the cell is expressing a halo-tag fusion protein in the plasma membrane, and it is also expressing a EDGHFR fusion protein in the cytosol. And when we treated those cells, mammalian cells, with a photo-caged dimerizer, so this photo-caged dimerizer has a NMO group and a chloroxyl group. This chloroxyl group will form a covalent bond with the halo tag, and when you shine this 405 nanometer blue light laser, this red thing will fly off, which is the NO group, which allows the exposure of EDGHFR, which is diffusing in the cytosol. It gets recognized to the trimethoprim, and it gets recruited. Since we use this TIRF microscope, total internal reflection fluorescence microscope, and we have this yellow fluorescent protein, which is moving from cytosol to plasma membrane, we could record the intensity change. And also we were targeting RAC GTPase, which is involved in the cellular motility. We can see if we can perturb cell motility, cytoskeleton, etc. So I just want to show you a microscopy image of what I'm doing here is I'm shining light, a shining blue light, and in a confined spot. By shining the light, I'm able to uncage this NO group. Thereby, I'm able to recruit the cytosolic YFP EDGHFR RAC protein. And because of that recruitment, you can see the cell is moving in one direction. And the movement is through lamellopodial protrusions. These are neuroblastoma cells. Just to give you a snapshot, so I flashed the blue light 4 times. So you could see the intensity increase in the respective image. This is photoactivation. And it happens only in the region where I shined the light and not in the region where I didn't shine light. There was no intensity change. So we did a chymograph to confirm that this is a site-specific. And also we did some quantification with respect to the cell protrusion velocity in the leading edge and the trailing edge. So why am I explaining all this to you is because this is one reason why Craig hired me in the lab. I convinced him saying that this thing, this photo-caged dimerizer without the NO group, it looks like a heterobifunctional molecule. And he made a similar story with the halo PROTAC, and the instead of trimethoprim, if it was a VHL ligand, it was halo PROTAC, and I was able to convey that okay, this is heterobifunctional, and that's how I ended up in Craig's lab. When I joined Craig Lab, I got that exposure. I was given an opportunity to work with molecular glue, and I came to know about this story before, but I got into the details when I was in Craig's lab. So thalidomide is a drug in the 50s. Early 50s, it was prescribed as a safe sedative in pregnant women who had morning sickness. So, and this is the structure of thalidomide. But then what happened was a tragic event is that this widely prescribed drug turned into a global tragedy. So it's basically, it led to more than 10,000 kids with birth defects, phocomelia. They don't have this limbs, upper limbs. So, and it took them a while to find that it is connected to thalidomide. And although in the sad part, this also made the drug evaluation process rigorous. And for that reason, Frances Kelsey, who was at the FDA at the time, she was preventing thalidomide getting into the US because she was not happy with the data what was provided by the company. So, and she kind of prevented this event happening in US, and for that she is treated as a hero even today. And if you want to know more about the legacy of thalidomide, you should read this book, Dark Remedy. So the thing is, although it is very tragic, in it the thalidomide has some anti-proliferative property. So it was given as a black box drug for leprosy and also multiple myeloma in the late 90s or early 2000. But it took us, the scientists, 5 decades to find what is the reason why thalidomide was causing this teratogenicity. For people who don't know what is teratogenicity, it's a structural and functional defects in the fetus or embryo during pregnancy. So the scientist from Japan, Hiroshi Anda, so what he did a pull down experiment. He conjugated the thalidomide with beads, and he immobilized the column with thalidomide, and he was able to beautifully pull down cereblon. And he found cereblon is the protein that is thalidomide interacting with, and he connected the dots, and he said cereblon is doing something, and probably that is the reason why the teratogenic effect is happening. And he also did some animal experiments. This is in the zebrafish. When the zebrafish is treated with thalidomide, you could see the limb defect, which is kind of mimicking what we saw in the newborns. You could see that limb defects with the increasing concentration of thalidomide. On the other hand, in chick embryos, when the same experiment was done without thalidomide and with thalidomide, you don't see a defect here, you see a defect. But also, when you mutate the residue of cereblon from tyrosine tryptophan to alanine, that defect got restored. So he kind of confirmed that it is the cereblon which is involved in the teratogenicity effect, and that leads to the limb defect. So that story was already exciting, and in the early 2010-ish time, it was people were also seeing using lenalidomide. It was having anti proliferative effects in multiple myeloma and also in B-cell hematological malignancy. So lenalidomide is a derivative of thalidomide. So the moment Hiroshi figured out it is cereblon, which is what thalidomide was binding to, and Professor Benjamin Ebert's lab, they also thought it could be cereblon, but they don't know what is it doing. And what is so basically they thought it could be a they found using quantitative proteomics, they found that it is doing something with these two proteins, Icaros and Aiolos, IKZF1 and IKZF3. These are zinc finger transcription factors which are very difficult to target and drug. So they found that lenalidomide is able to recruit Aiolos and Icaros and ubiquitinate them and degrade them. So this degradation is what bringing you that anti-proliferative effect in multiple myeloma. So this is great. Now you make a small change from thalidomide to lenalidomide, you are able to create new drug. But still the missing piece is, okay, what is happening? Why is the teratogenicity happening? So that puzzle got finally solved by Professor Eric Fisher along with Professor Benjamin Ebert's lab. So what they did proteomics here, mass spec. So what they found is there is a target called SAL4. It's a transcription factor. So they found that when SAL4 is degraded, so there is a limb defect. And what they did is it was they found in patients DRRS. It's a Duane radial ray syndrome, which is similar to this thalidomide embryopathy. So they saw the phenotype match and they were able to confirm that SAL4 is the reason. So what is happening is this thalidomide binds to cereblon and recruits SAL4 and degrades SAL4, and SAL4 is necessary for the limb development. And because of the SAL4 degradation, thalidomide was causing the teratogenicity. And now people started expanding this analogs of thalidomide from lenalidomide and pomalidomide. And you can see in this heat map, the substrates got increased as well. And some of the substrates are zinc finger proteins. Zinc finger proteins are very interesting for pharma companies because these are transcription factors, as I said before, and they are very hard to target. If you come up with a glue which could degrade a transcription factor, it is very attractive even now. So keeping all this in mind, what my boss, Craig, did is he first created a thalidomide library. So we made like 1130-ish derivatives of thalidomide. And with that thalidomide library, we want to see if we could find new targets, new molecules to cure different diseases, not just cancer, but also other disease. So there were two approaches. The first approach, we made the library. We didn't make, we got it from a CRO. And then we subjected them to PRISM screening. It's like hundreds of cancer cell line will have this thalidomide library, and you will get to know which are the cancers that respond to that particular derivative of thalidomide. You pull all that data, you will know there is one compound which is killing so many cancer cells that's attractive. And we also did L1000 profiling. So the thing is, while doing this in the lab, we found so many targets that were degrading GSPT1. And that was not attractive because so many pharma companies were after it, and they had the composition of matter patent for GSPT1 along the space. So Craig was not interested in GSPT1. So he said, if there are molecules which are killing wide range of cancer cell line, but it is not hitting GSPT1, I am interested in that. So if we were doing all the PhDs, postdocs, research scientists were given some molecules and we were doing Western blots and we were checking if it is hitting GSPT1 or not. And if it is not, then we were finding the targets and we validated the target. That's one approach. On the other end, because these are all zinc finger proteins, particularly C2H2 zinc finger protein, and particularly there are criteria to target, like why certain image targets certain type of zinc finger transcription factors. So we found from literature what are the other interesting zinc finger proteins we want to target. We fused them with nanolook and we had a control firefly luciferase plasmid, and we created a stable cell line. We treated those thalidomide library with that stable cell line. And then we were measuring the ratio of the luminescence to see if there are any degraders. And if there were any degraders, we can't be greedy. So it was not a clean 100% degrader. It would be 10, 20, 30% degrader. If there was any structural activity relationship between the library, then we took it seriously and we went further. I was not having this slide till yesterday, but I got this paper. This paper is available online, so I can talk about it. So from that library, one of the hit what we got was for this target LRF. It's ZBT-B7A. It has 4C2H2 zinc finger motif. And the thing is, the story is like this. This was done with Professor Gerd Blobel in CHOPS, Philadelphia. So the story is like this. Sickle cell disease is a big problem and also beta thalassemia. So the thing is fetal hemoglobin can be a good therapy for a CD and beta thalassemia, but the current options are limited. So the current option is hydroxyurea, which has a different response in different patients, and also it's not very efficacious. The second therapy is gene therapy. Bluebird Bio and CRISPR Therapeutics, they are cost around $2 to $3 million. So Craig thought, okay, maybe if we could solve this problem with a small molecule with a glue, that would be fascinating. So what was found in biology is there is this protein ZBT-B7A. It represses the fetal globin genes. So if you degrade this protein, you would increase the globin content. That is what he initially proposed. And then from that library, we got 3 molecules. They were structurally similar. They were showing 20 to 30% degradation of ZBT-B7A. And then this Curly from our lab, he made so many molecules, 70 to 80. He was optimizing the molecule to have less off-target effect. It should not degrade icarosilose. It should just degrade ZBT-B7A. So from this library, was able to get this molecule. And now probably Craig is thinking if we could do something with this. He might have already started the process. So by degrading LRF, which is ZBT-B7A, we could activate fetal hemoglobin, which could be a therapy for sickle cell disease. So why molecular glues are booming right now from the academic lab to industry. As I said, thalidomide was approved, withdrawn, approved for multiple myeloma. Lenalidomide was even approved for other hematological malignancy. Now we have pomalidomide, which is even more potent, is used for relapsed refractory myeloma. So the thing is, because of the undruggable proteome, it's a well-established mechanism. So the investment is crazy right now. So within the last 3-4 years, there were more than $15 billion invested in development of new molecular glues. And also 3 months back, Craig started a company, Quarithera, and which received attention and also significant investment. So I'm going to change gears here. Although we discussed molecular glue, Craig started PROTACs. Craig is very exploratory. So the lab also did something called regulated induced proximity targeting chimeras. It's called RIPTACs. The reason why we are talking about RIPTACs is Johnson & Johnson a few months ago acquired ALDA, which is the licensed entity of RIPTACs. They made the RIPTACs. It got acquired for $3-ish billion. So it became a sensation around. And we are going to dig deep around this data. So first, what is RIPTAC? So this molecule HLD-0915 is it's a heterobifunctional molecule, very similar to my PhD work. And so what it does is it has ligands. One end, it binds to the tumor-specific protein, and the other end, it binds to an essential protein. So in this case, it's BRD4, and the tumor-specific protein is androgen receptor. So their lead indication was prostate cancer. So that's why androgen receptor. So what happens here, the moment you add RIPTACs, they are small molecules, they can penetrate the cell, they can also enter the nucleus. So they form something called ternary complex. And this, because of the ternary complex formation, so there is an inhibition of the BRD4 because of the ternary complex formation, which leads to the cancer cell death. So it's not like there are BRD4 inhibitors already in the market, but they are very toxic. Whereas here, you need the stable ternary complex formation to happen for the inhibition to occur. So because cancer cell express this androgen receptor in excess, so you have a very broad therapeutic window. So which is what I'm explaining here. So there are healthy cells which has the essential protein, but the RIPTACs do bind there. But for the inhibition to happen, it needs that cancer-specific protein. So then this ternary complex formation leads to the inhibition. There is apoptosis trigger. And this therapeutic window is one of the reasons why this molecule is very efficient, I would say. So I just want to touch base on the how they took off. So what they did, they made a very simple cell line development. So with 293 T cells, this green one, they were just GFP expressing 293 T cells, and the blue one, they are halo tag FKBP fusion, a stable cell line. So they are expressing halo tag and FKBP. So because they express halo tag and FKBP, so they could use chloroalkane, which binds to the halo tag, and they used FKBP, which is trimethoprim. So they were using trimethoprim for this one. So it's a covalent RIPTAC because the halo tag binds with chloroalkane in a covalent bond, whereas here the FKBP trimethoprim is non-covalent. So the thing what I wanted to say is that, so in the viability graph, you could clearly see for this different targets. So JQ1, it's a BRD4, and so there are other kinases and CDKs. So these are all ligands inhibitors for that. They made already existing inhibitors, or it was there in clinical trial, pulled off for some reason. They took those molecules, they added a PEG linker, and then they were adding chloroalkane and FKBP, and they were able to show that only in the FKBP halo tag expressing cells, they were able to see a strong effect. You could see the fold shift, which is the therapeutic window. So the RIPTAC has a enhanced anti-proliferative activity, and it is because of that halo tag and FKBP. So they were able to show it for different targets, and they were showing it for covalent and non-covalent. And one other thing, what they also did in that paper, they did multiple things. What caught my eye is it's very, the viability effects are target dependent. So in the presence of, although they were adding these molecules, along with that, they added the targeted molecule, which we don't know. We don't have access to the structure. So that molecule, the target effect you could see here, it was having a competitive inhibition. So it clearly means that it is very selective, and it kind of validated the differential biology, which Craig often stress, the word differential biology. It clearly mentions that RIPTAC works according to the mechanism what we think. So then from cells to animals, so they used mice models, different types of prostate cancer mice models, enzalutamide sensitive, enzalutamide resistant. Enzalutamide is the first line of treatment for prostate cancer. It targets androgen receptor. So here you could see the HLD, which is the red. You see tumor volume in the y-axis and the treatment days in the x-axis. You could clearly see that HLD is regressing. It's not allowing the tumor to grow, whereas enzalutamide is slowly growing here. It's growing even faster here because it's enzalutamide resistant model. Even in the sensitive model, it is growing, but it is not significant. And in this model, it is there. So both enzalutamide and ALDA molecule, they both show efficacy. So take home message here is the ALDA molecule, irrespective of whether enzalutamide sensitive or resistant, is showing superior efficacy in reduction of the tumor volume. So whatever data I present, I discussed it with Nilesh. Nilesh is the clinical ops from ALDA. He couldn't make it today, or else it would have been even more interactive. So the thing what we see here is, so they took patients, 31 patients. Out of them, 22 of them received more than 2 cycles of therapy. Each cycle is 21 days. So the therapy is the ALDA drug. So here you have 21 patients. So they underwent different doses, 12.5, 25, 50, 100. And in that, they saw some effect. So what are we measuring here? We are measuring the change in the PSA, prostate-specific antigen. And if the patient reported less than 50% of PSA from day one, so it's PSA 50. If they have a dip more than 90% in PSA, it's PSA 90. So what we see here is at the dose of ALDA drug at 25 mg and 50 mg, there was people were able, ALDA was able to see significant drop in the PSA 50 and 90. So they thought, okay, this is great. The PSA is coming down, which means the patients are responding, and we should go after 25 and 50 milligram as their RDI dose final dose. So here I add these questions: is it responding only for certain type of androgen receptor? I got this information. So partially we have data about the genetics of the patients. So some of the patients have AR which is amplified. Some of the patients have mutations in the binding domain. Some of them have mutations in the tumor suppressor gene. So we have the data. So the take-home message: the drug works in wide variety of population. Even if you have all of them, you see a response. And there is also, they measured circulating tumor DNA as well, and it was going down significantly. So I'm just going to show some pictures just to prove the point that, so this is a CT scan. This is a CT scan. And what you see here is the patient. This is a patient number, and they took this 25 milligram drug, and this was the image taken on day one, and this is the peritoneal mass, the tumor volume. And after 27 weeks, 10 cycles of the ALDA drug, the volume got shrinked. That is what they were able to show. And so there is a reduction in the prostate specific antigen. There is a reduction in the circulating tumor DNA. So they were able to report significant tumor reduction at different cycles. So this kind of conveys the message that it is able to do the tumor reduction successfully. This is the money slide to me, from my point of view. So this patient is showing unconfirmed partial response. So it's not curing the tumor. There is a tumor reduction, I could clearly see. There is a reduction in the PSA, and also they didn't have data with the circulating tumor. But then the tumor volume is reducing significantly. So this patient who has this prostate cancer was having so much difficulty to urinate. So when the drug was given to this patient, after a few weeks, he was able to urinate, and he gave that the comment that he felt like that his cancer is gone. And he was able to feel it, and he was able to urinate after taking the drug. And according to that, from that comment, I think ALDA was very proud that they made this. They were able to come up with this drug, and improve so many patients' life. So my conclusion slide from the ALDA's presentation is basically, this drug is given once a week. It is well tolerated. Someone even asked about the adverse events. So I was surprised. There was 0 adverse event for one of the doses. But then it was a small population, 13 people or something. And there was a strong PSA reduction, more than 50% reduction in PSA at all doses. And the 90 responses were happening in 12.5, 25, and 50. It was not happening at 100. I asked him why. I can speculate why it might be, but I don't know the answer. So most of the patients showed response. the partial response. So when you say full response is like when the tumor is completely gone, then you say full response, complete response. So the activity worked both in AR and non-AR genetic alterations. So they selected this 25 and 50 milligram. Right now, they are doing the next level of trial because now they are acquired by Johnson and Johnson. Johnson and Johnson will be doing all that part. So the thing is, this drug is working. It is safe. It is having very less side effects. So with that, I would finish my talk. I just want to thank everyone, thank Nathan and Christian and the DDD team for the opportunity to speak about molecular clues and RIPTACs. These are some of my favorite books because this is a pulse challenge for postdocs. I just thought you should probably give it a read. It is very inspiring and motivating. Thank you.